The overall goal of this method is to genotype complement receptor 1 length polymorphisms for use in the assessment of susceptibility to diseases such as Alzheimer's disease to better understand the role of CR1 isoforms in disease pathogenesis.
This method can help answer key questions in the neuroscience field, such as the pathogenesis of Alzheimer's disease. The main advantages of this technique are that it does not require fresh blood samples, and it's easier, cheaper, faster, and therefore applicable to larger populations.
We first had the idea for this method when we recognized the challenges in performing Western blots regarding the necessary time and need for fresh blood samples.
Pipette 20 microliters of proteinase K into the bottom of a 1.5 milliliter tube. Then, add 200 microliters of sample to the tube. Add 200 microliters of lysis buffer to the sample. Mix by pulse vortexing for 15 seconds. After mixing, incubate at 56 degrees Celsius for 10 minutes in a water bath. Briefly centrifuge the 1.5 milliliter tube to remove the drops from the inside of the lid. Then, add 200 microliters of ethanol to the sample. Mix again by pulse vortexing for 15 seconds before briefly centrifuging the tube as before. Next, carefully apply the mixture to the membrane column in a 2 milliliter collection tube. Without wetting the rim, close the cap and centrifuge at 6000 times G for 1 minute. Place the membrane column in a clean 2 milliliter collection tube and discard the